I introduced you to different kinds of algorithms, almost like an introduction to these algorithms. And now I'm going to take a different direction as segue into an introduction into biological databases. Given that you have been doing these labs and you have been accessing these databases, and there have been questions about it. So I'm going to talk about biological databases and why they are important and how one navigates through these. And then after that, I'm going to go into a little bit. I have. I'm going to sort of talk a little bit more about the fourth module about technologies in bioinformatics because there seems to be a lot of interest. And this is actually some parts that I'm taking from the genomics and the proteomics course because a lot of you actually expressed that you will not be able to get to do that course. So I'll take the best bits of that course and actually add it here. And then in module 5, so we're also going to have the quiz in that week. So just to let you know, so the quiz will be covering everything that has been taught till the day before the quiz. The quiz is going to happen on Friday in that week. Um, and yeah, and then fifth module, I'm going to come back to the algorithms. And specifically, then we're going to look at how do we sequence an entire organism. All right, so going to the module today. I hope everybody can see this part of the screen in biological databases.
Yes, sir.
All right.
Yes, sir.
So I'm going to first start with a historical perspective. Then I'm going to talk about NCBI, the PubMed website that you're already looking at. And then I'm going to talk about EBI and Ensemble, which is a different type of aggregation of data. So the growth of bioinformatics, as I have already told you about, is essentially going from initial molecular biology experiments that was simple, and they were usually you do an experiment, you have a finding, and you report that finding, right? And these findings were typically consisting of discovery of molecules and machineries that was involved in the process of DNA replication, genome replication, transcription, synthesis, and gene synthesis and protein synthesis, right? But with the first exception came with the discovery of reverse transcriptase, which is basically an enzyme in viruses, which allows them to copy RNA into DNA. So this whole process of the central dogma was reversed. And after this technology or after this discovery, a lot of new technologies started to come up whereby they could actually now, um, after identification of these molecules and machineries, they started to actually diversify, or they started to identify, characterize them across the entire animal kingdom, animal and plant, across the entire life kingdom. So these molecular sequences which came about had to be stored, they had to be readily accessible, and therefore you had databases or data banks developed for this. So when the above completed algorithms were developed, like when you had a lot of information, then algorithms were developed to identify specific features within these molecules. So, for example, when people sequenced genomes of organisms, then they wanted to identify where are the genes in those genomes. And once they had identified where the genes are, then they would like to identify where is the open reading frame in those genes, where does transcription or translation start and stop, so on and so forth. All right. So difficulties remained in how to distinguish, still remain actually, in how to distinguish short open reading frames and actual protein coding sequences. This is still a problem which is not satisfactorily solved. But one thing which is common to all these databases, as you would have already started to appreciate, is that they depend on bioinformaticians' ability to compare sequences. And typically, when you're comparing sequences, you align those sequences one against the other to basically juxtapose them. You put them next to each other, and then you either are able to compare them one base at a time, or you are able to compare blocks of nucleotides or amino acids in those sequences. And from those, you can actually, from these kinds of alignments, you can start to, I'm sorry for that. Yeah, and from those alignments, you can actually infer things like structure, function, and the relationship of proteins. And this entire course, actually, as you would see, is built around how we do these things using different kinds of algorithms. So specifically, the sequence alignment algorithms are something that we're going to talk about in the second half of this course after the minor. All right. So given that there is flooding of data today, you require automation, you require faster ways of solving these problems. So these are some of the early
Sir,
databases and algorithms that were developed. So databases
sir,
are in purple. Yeah.
sir. What is the difference between gene and ORF? I wanted to know, sir. I
So a
think I have.
gene.
Yeah.
Okay. You want me to answer or do you have something else to say?
Yes, sir. You please answer.
Yeah. So a gene basically is inherited, whatever. It's a location where you would be having from where you would have an mRNA transcribed and a protein will be made from that mRNA molecule. Okay. Now, the gene itself is a location on the genome, right? Which will
Yes, sir.
have the which will have things like the Tata box,
Yes, sir.
which will have certain things like your transcription factor binding sites. The gene will also have a transcription start site and transcription end site. All right. But within
Yes,
this gene, the part which actually codes for the protein starts from the open reading frame. So it's called the open reading frame because that is where you will have the first start codon present in the gene. So you will have an AUG present at the beginning of the open reading frame. And given that you have a AUG, then every subsequent three-letter nucleotide would correspond to the protein sequence or the
yes,
amino acid that comes in the protein sequence. Now, because let us say, I'll just give you an example. Just imagine that this first line is a genes sequence. Okay. And
yes,
each
sir.
of these three letters is corresponding to an amino acid, right? That's how codons work, right?
Yes,
But
sir.
if we do not know at what point of at which of these bases the actual translations have starts, right? So let us assume that translation starts at A. I'm just giving you an example, right? Translation actually starts at AUG. So let us say translation starts at A. So when you are given this gene, you don't know where the translation starts, right? Which does it start at this A? Does it start at this A? Right? So an open reading frame actually tells you where does the translation actually start and till what, how many bases does the translations go on. So let us say if this first line is a gene, we say that the open reading frame starts from the A in the word atlas and it goes to T in the word proteins. So although you have this entire gene, when a mRNA molecule is going to be made, it's going to be made from all the way from D to S. But the part which codes for the protein is only going to be from atlas to T to prot.
Yes, sir.
There are these parts of the mRNA molecule which do not code for the protein. They are called untranslated regions, and they have certain um, uses in the cell. And if you are more interested into them, I can give you some reading material around it. Okay,
Yes,
so
sir.
open reading frame is that part of the gene which codes for a protein. The gene is the entire gene is basically a location that contains the open reading frame, but also contains that part which gets transcribed into an mRNA molecule. Okay,
Yes,
so
sir.
so
And
here is
which
an
not
example of
and which is not transcribed, like regulatory sequences also considered in the gene, a promoter
regulatory
region.
sequences. Yeah, so regulatory sequences, no, regulatory sequences are not considered as a part of a gene. No,
Like operon, like first comes the
operon is different.
upstream to the
Gene is different.
transcription start side, there is lac repressor, lac operator, and then there is what is called uh, that
So I think yeah. So maybe I can complete what you were trying to say. So when you are talking about operon, Pratnesh,
Yeah.
operons are having multiple genes.
Multiple
And
genes,
operon,
yeah,
and
multiple genes or
yeah.
multiple open reading frames, like they have only one. uh,
So if you will have multiple genes, you will have multiple open reading frames by default,
yes sir,
right?
but multiple
Yeah.
genes may have
So let me
multiple
complete. Let me
transcription
complete.
start
Let me
site.
complete. Let me complete.
Yes sir.
So when you talk about lac operon and you talk about the binding sites, right? Inside the operon, you would have the binding sites, but inside the LAGZ gene itself, the location point from which the gene starts to transcribe and the location till which the gene transcribes, that will not have a LAGZ bind, whatever, LAG I site or one of those repressor element
Yes.
sites. Those repressor element sites are typically going to be outside the region which is getting transcribed, right? Because the repressor is going to come and sit there, right?
Yes sir.
Or the transcription factor is going to come and sit there. And once it
Correct.
sits there, then it stops the transcription of the nucleotides which lie downstream of it. So typically, these regulatory motifs are not a part of the gene's definition. But yes, when you talk about an operon, which can contain multiple genes, and each gene can actually contain multiple open reading frames as well. Now, don't ask me what that means. Just take my word for it. If you want to know more about multiple open reading frames per gene, we can talk about it outside the class.
Yes sir.
But
Yes.
yeah, typically each gene has at least one open reading frame, which tells that from this location to this location in the mRNA molecule that is going to be made from the gene would be coding for a protein. Okay, so coming back. So these are the few databases and corresponding algorithms that were developed to work on the data in these databases. So the first databases were database of proteins. So this was the protein data bank. And then correspondingly, you had an algorithm called the global alignment algorithm, other words, Needleman-Wunsch, because these are the people who wrote the algorithm. Then in 1981, you had the local sequence alignment algorithm. So these two algorithms are something that we're going to talk about in modules 6 and 7. Then you had the gene bank database that was created in 1982. After the gene bank database, you had the FAST-P algorithm that was basically used to search for protein databases. Then you had the Swissprot database. Then around late 1980s, you had the establishment of a new programming language called Perl. So before Python was in fashion. So for example, when I was in college, it was all about Perl because Perl was quite handy, but it has certain limitations. The libraries people did not keep up with Perl in terms of library development, and Python is much faster than Perl. So Python has actually taken over from Perl. Then in 1988, NCBI was established. That's National Center for Biological Information, which sort of was a single stop area for combining all these databases and also deploying a lot of these algorithms through some of the tools that you've already seen during your lab courses. So the FAST-A program was released in 1988, where you could actually search for DNA and protein sequences if efficiently in databases. Then in 1990, a really big event happened in terms of algorithms. The BLAST algorithm was released, which allows for much faster quick database searches. And around 1985 to 1990, that is where the Human Genome Project was conceived, and there was a host of technologies that came about. Now, looking at this figure, can somebody tell me intuitively why did we start first with protein databases?
Sequencing of protein was available first, before
Sequencing of
the
proteins was,
sequences of DNA.
yeah. So that's correct. So can you guess why?
Like protein sequencing was developed by Sanger, Frederick Sanger in 1956, and that Sanger sequencing for DNA came into 1977. So when it comes to 1977, then how come that data bank be available?
That's good.
Like
That's good.
that's why
But I'm
protein data
saying that
bank is
how would
available.
you how does one sequence proteins? Any ideas? Do you require
I don't.
enzymes or other things to sequence proteins? Or is it a much simpler thing to do? Anybody wants to give it a try? You have done the method already. You have actually done your laboratory practicals in previous courses
As
using the
now we
machine.
are doing, we have not ever done.
Huh, I mean, at least on paper, you have been exposed to that machine. So protein sequencing, so Ratnesh is right here that earlier it was available. Sequencing proteins was easier because proteins are typically much smaller. First, second, it's easier to isolate proteins because proteins are can be in specific locations inside the cell. Whereas on the other hand, DNA, all of the DNA is located in a single location. It's much harder to isolate at that point of time a single gene until PCR was invented and until all these enzymes were found which could cut and paste DNA. Proteins are simply polymers, so you can take a protein like insulin, which was used in from early 1920s to treat diabetic patients. So you could take a protein like insulin. Um, you could acidify the bases so that you break the polypeptide bond. So the C bond between carboxyl group, that is COOH, and the amine, which is NH3. So that you break the CONH bond by acidification reaction, and that led to basically each peptide come out of a protein sequence, and that peptide could then be seen in a LCMS, a liquid chromatography mass spectrometry instrument, and you could basically tell what is the composition of amino acids in a protein. So I hope everybody has been exposed to the LCMS machine at least. Show of
So
hands,
I
please.
don't think so.
Do you know what an
No, sir.
LCMS is? No? Okay. Okay, so LCMS is basically a very sensitive uh, very sensitive weighing balance. So you can actually measure atomic weights. So you can actually distinguish between atoms and molecules in a LCMS. So just imagine it as a very sensitive weighing balance. All right. So when the Human Genome Project began, there was already some existing in work that was going on that led to the project, and this is the timeline for these projects. So right from around 1965, people started to actually sequence once we had a lot of these enzymes available, such as restriction enzymes that could cut DNA, DNA ligases, which could paste DNA, and the advent of cloning, which could take a piece of DNA, put it inside a DNA vector, and that vector gets cloned or gets transformed in a bacterial cell. And as a bacterial cell grows from one to millions, the copy of the vector also grows from single or a few copies to millions of copies. And as a result, the DNA molecule that you were able to cut and paste inside a vector also increases in size. So you can see that a lot of this sequencing earlier was for much smaller fragments. So the first sequencing happened for the partial tRNA fragment in yeast. Then you had the 5S rRNA in from E. coli that was about 120 bases. Then you had around in 1975, you had between 1975 and 1980, you had sequencing of the MS2 phage, which is an RNA phage, and then you had the sequencing of phage 5X174, which is approximately 5,000 base pairs. And then around after 1980, there was a big explosion of in the size of the sequencing, uh, temporarily primarily driven by the advent of Sanger sequencing, which was published in 1977. And there was another technology that after Sanger sequencing, because of which there was another jump in the size. So from about 170 kilobase pairs of size after 1990, the researchers went to one order of magnitude further to hemophilia influenza. That's the first genome that was first genome of the bacteria that was sequenced. So can somebody tell me what could have happened between 1985 and 1995 that could have pushed like one order of magnitude more
So blast.
in terms of, so BLAST is for searching. What other technological advancement happened during this time?
Shotgun sequencing.
What kind of sequencing?
Shotgun sequencing. Oh,
So shotgun sequencing was already happening at this time. So yes, it was sort of shotgun sequencing driven, but what was one very simple technique which actually
PCR.
allowed exactly. So around 1985, the PCR technique was developed, which allowed you to amplify a piece of DNA multiple times. And this is a very simple technique, but it's very, very powerful. Even now, we see PCR is affecting all of our lives. All right, so this is the timeline for DNA sequencing, and typically around 1990, that's where your Human Genome Sequencing project was conceived, and a lot of technologies around the project were developed. So they were like, we'll talk about the project, but you can see that since the Human Genome Project, lots of technologies were came up, and going from sequencing single different kinds of organisms. Now we are in the era around 2010s. We were in the era of sequencing many individuals of the same species. So, for example, the first was the 1000 Genome Project, which was completed just around 2010s. Then we had the 10,000 Genome Project that was also conceived around this time and began. In 2015, America launched what's called All of Us project, where they want to sequence about 1 million human genomes. If you are following science news, there is something called the Earth Biogenome Project, where they want to sequence every vertebrate organism on Earth. And around in 2020, uh, India also started played a major role, and we have started what's called the Genome India project, where we are going to sequence 20,000 Indians from different regions because of our unique geographical history, and IIT Delhi is a part of this project. All right, so this is the timeline of DNA sequencing, and in the next class, we are going to actually dissect the technologies that helped us to basically go there. But you can clearly see that from very small genomes for very few organisms, we have very rapidly expanded into much larger genomes for many organisms and many copies or many individuals of the same organism. So this clearly requires much more efficient ways of storing data and of analyzing that data. So biological databases essentially store sequences. All right. Similar biological sequences tend to be related, right? So what kind of information they are storing? They are storing functional information, structural information, and evolutionary information. Just give me a second. Sorry for that. Yeah, so here's a list of databases which exist according to one of the latest publications published last year. So there are already around 1,500 online databases, and they range from different kinds of molecules that they target. So, for example, there are nucleotide sequence databases, there is RNA sequence databases, there are protein sequence databases, to information about those molecules, such as structural databases, genome databases, metabolic signaling pathways. Then there are conglomerates. So there are databases of collection of genomes, such as human and vertebrate genome databases. There are databases around specifically around genes and how they relate to diseases. Can somebody tell me the name of one such database that you have worked on in the lab, last lab?
OMM. OMM.
Yeah. So OMM is one example. Then you have databases to store information of experiments, specifically experiments around gene expression, because your DNA is the same, but the expression of that DNA or the number of molecules of different RNAs that are getting made from those DNA is different. and these are stored typically in microarray and gene expression databases, right? And similarly, you have proteomics databases as well. So there are lots of databases you can see, and they are growing in number. So can we come up with a sort of formalized definition of the kind of databases that exist? So there are databases which are called primary databases. So this is where experimentalists submit original data. So they submit raw data. What can you submit here is controlled by the submitter, right? So if I am an experimentalist and I generate data, I can submit this primary data, and this is controlled by me, what I'm submitting. And this submissions can go in databases such as GeneBank, which is a database of genes. These submissions can go into databases which stores variation of new differences of nucleotides between humans. So there is a database called dbGap, which stores the information of how frequently specific locations in human genome are mutated between people from different parts of the world, right? So typically name, yeah, so here I will, it is sort of a homework problem. Can you find out the name of a primary Indian database where microbial strains are deposited? So this should be simple for somebody who is working in probably, say, Professor Preeti's lab, or has been exposed to work from Professor Saroj Mishra's lab, who was in the department until a year and a half ago. So this is a
So
homework
it
problem.
is in Pune or in, I guess, in Haryana, like microbial depository, something called. Is it?
Yeah.
Are you talking about that?
Yeah. So please find out what the name is. And this is a sort of a homework thing. The other databases which are there existing are called derived databases. So these are databases are built on primary data. So this is basically that if you have submitted some data, somebody does the analysis, a bioinformatician typically does the analysis and creates some meta information about the data, and this is typically controlled submission. So for example, if you submit some data as a primary experimentalist, somebody sitting in the US might analyze that data and can come up with certain information about it, but that can be also bogus information. So particularly this kind of databases are controlled by a third party, and third party, for example, the NCBI. And there are examples of this. For example, RefSeq. Can somebody tell me what does RefSeq do? What information does RefSeq have? Now you have probably looked at it.
About proteins database?
Anybody else wants to give it a try? RefSeq was mentioned in the last lab. Okay, so RefSeq essentially is a database where you have reference sequences. So RefSeq tells you for a specific version of an organism's genome, let us say the human genome, how many genes have been annotated in the human genome, what is the sequence of the human genome, from what location in the chromosome to what location in the same chromosome does a gene lie, so the start and the stop, physical location. What are the identified oris, for example, in the genome, where do they lie in the sequence, so on and so forth. All right, so there are essentially two types of databases, primary and derived. There's also a third kind which I'm going to come to. All right, so again and again you will have to hear about this word NCBI, which is essentially National Center for Biotechnology Information. It's uh, located here in the state of Washington, DC, uh, in the US, and it was established in 1988, and it's a part of National Library of Medicine, at National Institute of Health, which is basically the central governing body in the US. So its job is to establish public databases, do research on computational biology, develop softwares, disseminate the information around all these tools that they have developed. Okay, so one of the first databases that NCBI actually started to sort of started to talk about or disseminate was the gene bank. So gene bank is NCBI's primary sequence database. So this is where you will get information of nucleotides, nucleotide only sequence database. It has archival in nature. So basically, if you have submitted the data and that data is not being used for a long time, so it gets archived, but no data is lost. And even you as a experimentalist here in this class, if you were to ever sequence something, you can actually submit this data into the gene bank, and you will get an accession number associated with your submission. That is, you can actually pull out the data, and they will be responsible to host that data and make sure that that data is never lost. You can do direct submissions, so basically in a click wise fashion, or you can do batch submissions where you can submit lots of data simultaneously together. And this thing was also facilitated through a file transfer protocol that's also called FTP accounts. So there are three major collaborating databases which forms the NCBI's primary database. One is the gene bank, one is the DNA database of Japan, also called DDBJ, and the other one is called the European Molecular Biology Laboratory Database, or the EMBL database.
Sir, I have a
This is
question.
how, yes.
Sir, sir,
Yeah, please go on.
genome sequencing is still considered a big thing. Like I was saying, like 3-4 months ago, somebody from IISER Bhopal, people have sequenced genome of Giloy that we use nowadays. So it was published in like most of the newspapers. So is it considered like sequencing is just still considered as a big thing in India at least?
So when you say big thing, it's so I think it grabbed news because it was Giloy itself. So it's a herb, right? And Giloy has been shown to reduce fever. Specifically, it was being prophesized as the miracle drug in COVID. So I think that is why it captured attention. But yes, a lot of the herbs in India, a lot of the Ayurvedic herbs, a lot of the naturopathy herbs, a lot of trees, plants, animals in India are not sequenced. And that there are big projects going around this specific effort. The team from IISER Bhopal, actually, I was in IISER Bhopal before joining IIT. They've also sequenced tiger, the Indian tiger, for the first time. They sequenced the peacock. They've also sequenced ginger, which is like native to India, and they're also sequencing some other organisms like dolphin and other things like that. So these are, yeah, it's when you say it's a big thing, I mean it's not a small thing, right? It's almost like it's a natural heritage. These organisms, which are uniquely found in our country. Okay,
Yes,
so
sir.
the
One
international
thing more. That they have
what?
submitted like it was published in bioarchive. So sir, is there some, so sir, why but
So
it was
bioarchive.
published in a yeah, why
So
it was
bioarchive
published
is
in a
basically
journal like that?
So bioarchive is essentially a repository of manuscripts. So when you have write a research article, you can either get it peer reviewed or you can submit it to an archive. Typically, when you write a paper and it goes under peer review, it takes a lot of time. So when you want to actually quickly announce what you have done, people typically put the paper which has not been peer reviewed by other people. So the work has not been checked by other people. So you can put your work in archive. This archiving for publications was started by physicists who are typically like putting their work on archive. So this work is put in our bioarchive, but that does not mean that this work is published or you should believe everything that is written in it. The other thing is why the genome was published. You probably would have to go and read the paper, because the paper is not only talking about the fact that they have sequenced Giloy, but they are also talking about a lot of interesting things about the genome of Giloy, specifically about the unique metabolic profile or metabolites that Giloy might be having that they can use to actually infer the medicinal properties. Okay. All right, so I'm going to go back.
Yes, sir.
If you have questions regarding what I'm teaching, Ratnesh, I hope please you focus on this and we can ask the other questions in MS Teams. I'm always answering them.
Yes, sir.
So the NCBI Gene Bank, or this NCBI sequence collection database, is again a collaboration. So it's NCBI, you have from EMBL, It's European Molecular Biology Laboratory, and you have something called NIG. So, and it's based on these data banks, and each of these data banks actually have also related search tools like Entrez. Entrez is something that you were using for in PubMed when you were going. So earlier it was called Entrez, now it's called PubMed. Similarly, in the case of EBI, if you go, there's their search tool is called SRS. And in the case of NIG, the tool was called Get Entry. So this was a little bit more uh, old school way of getting things. All right. So NCBI and EBI are basically sort of similar kind of databases. And the main, so yeah, one question that one has to come up with is all of them do the same thing. So NCBI and EBI both have publications. So one question that I have for you again as a homework question is to find out what is the name of the publication database for NCBI and what is the corresponding name of the database for EBI. Both of them stored DNA. So what's the name of database for NCBI and EBI and protein, so on and so forth, right? So this is how the Entree system looked like earlier, and now it looks slightly different, as you guys have all seen. So when we specifically
So
talk
I have a
about,
question.
yes.
So why do they have to like why do they want to make 3 to 4 different bodies that handle like the same databases? Why can't they just have one unified body which handles everything?
So actually there is a fourth body now, which is called CNGDB, which is a body from China. So now can you understand why you would want to have a different body?
Yes, sir. Like,
Yes, sir. So but like
these are
can't they have like like they can nominate a panel from and like each country can send like few representatives to the panel.
So what is the largest international
So
body
UN.
that you know of?
UN.
UN, right? Does UN contain all the countries in it?
No, sir.
Right? So it's the same reason. It's about matching, aligning everybody's interests, and everybody. Now you will see the reason why this Chinese gene bank is a new entry is because you see that this earlier science was typically driven by America and Europe and Japan, and now China is also driving research, and they would like to have their own repository, right? So it's about all about controlling information. So the more information you can control, the more information you can, it's all about flow of information. So also one has to put resources, right? So for example, when you go to NIH, okay, you go to NIH, all of you have gone to NIH and PubMed for your first two labs. You go and you search for a keyword, right? And because you search for the keyword and a lot of information is pulled from a database and shown to you. Now this entire process is free for you, right? So somebody's paying money to host the data, to host the algorithms to search this database, right? So it's basically if anything is given for free to you, right, then you are the product. I mean, this is what like Google and all these Facebook people do, right? So this is free for you because one, by doing this, they can justify to control the information because they are making it accessible to you. But also, secondly, every time you are searching for something, your IP address is locked, right? Where what kind of information you are getting is getting logged. So in one way, it's some kind of a sort of a master surveillance network which actually knows what's happening in terms of science around the world. So this kind of power everybody would like to have, and that's why there has been no consensus body so far. I mean, there was a consensus body from Europe, America, and US uh, and Japan. Sorry, but yeah, now there is a new body, and I think literally now people are actually making silos for themselves. Europe is actually making its own set of databases, as we will discuss coming on. So it's a little bit more politics than science in this aspect. So for example, here you can
Right,
ask
sir.
this question. I can ask the same question about space agencies, right? Space is not colonized right now, right? Nobody owns space. But you have the Russian Space Agency, you have NASA, you have the European Space Agency. You have the Japanese, you have the Chinese, you even have the Indian Space Agency, right? So everybody wants to have their footprint in these key areas. And now, specifically coming from the pandemic now, you will see a lot of efforts are going into controlling this space and controlling the flow of information in this space. Okay. All right. So this is how the entree system looks like. So now specifically, if you look at Gene Bank has 18 different kinds of data that you can submit to it. You can have primary sequences, you can have rodent sequence, you can have other mammalian sequence, you can have other vertebrate sequence, invertebrates, plants, bacteria, viruses, bacteriophage, synthetic sequences, unannotated sequences. So for example, you got a piece of DNA, you don't know which organism it came from. You can have ESTs, something called express sequence tags. We're going to talk about it later on. There used to be something called patent sequences where people were able to patent genes initially. This is a very interesting story. When the human genome sequence was being sequenced, there was a company which was also a private company which was doing this called Celera Genomics. We're going to talk about it in the next class. Who tried to sequence a few genes, who tried to patent a few genes. The patents were initially granted, but then they were subsequently taken away. There's something called sequence tag sites. So these are basically sites which allow you to recognize certain plasmids or certain unique organisms. They're called genome survey sequences. So again, these are sites specifically in the genome which help you to survey uh, the composition of a community of DNA samples. So this is typically called meta genome sequencing based things. Then there is something called high throughput
Sir,
genome sequencing. Yeah.
about patents of genes, so it was retracted, like they didn't give the allow the patenting the genes, but
Yeah.
that cellular thing was happening after 1990, I guess. But before that, people were there, like a first genome, a first plasmid was, I guess, plasmid or bacterial strain was patented by Ananda Chakravarti in like 1970.
Ask your question. Ask your
Yeah, sir.
question,
Like
Ratnesh. What is the question?
before
Your
that,
question is,
so that's the question. Like, yeah,
so
the
I'm
question is
basically
like,
telling you
yeah.
so your question is that I am wrong that sequences are still patented. No. So there was a litigation that was passed in the US, which basically said that you cannot patent sequences specifically if they are naturally occurring. But now, because there is a whole new field in biology called synthetic biology, where people are actually exploring the entire nucleic acid space. So now actually completely synthetic sequences are getting patented. And we can talk about it
I
outside
think
the class, but I don't want to discuss it further in class. Okay, I have to finish
okay,
the lecture.
okay, perhaps.
So
Okay,
this
sir.
is how a traditional gene bank record looks like. So it will give you some kind of a location or locus. So this is more like an ID. It tells you what kind of record it is, how long it is. These many base pairs. It's an mRNA. It's linear. The last date it was updated, and it's an INV, right? So this is INV code. This basically means it's coming from an invertebrate. So some information about it, definition, accession number, as you can see, some keywords, the organism. Then you have the author, the paper from which this record was taken. The paper itself has its own things, authors, so on and so forth, journals, right? So this is how you would typically store information about a typical gene bank record. But now again, you see, yesterday in class, somebody asked me how is ontologies sort of described or what's the idea? The idea is that you almost have like a form that you have to fill, right? You have like a controlled way in which what kind of information goes for each record is pre-decided, and that gets populated based on each entry. Okay, so again, this just tells you how it works. So this is the locus name, this is the length, molecule type, division, and the modification date. So modification date suggests that you have the ability to change the record as well. So each record can have versions. So typically what you will see that uh, accession number will have versions. So for example, this is the origin accession number, and point two means there have already been two versions of this same accession number. So maybe if you go to this accession number dot one, it would have been updated before this date. Okay. This gives you the taxonomy of the organism, so on and so forth. So now we come to the idea of ontologies and taxonomy. So taxonomy, essentially, what is it? It's basically a way of specifically categorizing information. Let us say if you talk about an organism, we can go from kingdom to phylum to class to order to family to genus to species. All right. So if we were to talk about a dog, right? Specifically, this kind of a dog. A dog would be from kingdom Animalia. It would be from phyla Chordata. It would be a It will be a mammal. It's a carnivore. The family is Canidae, so the family of all kinds of dogs. So from the family of dogs and wolves, Canidae, you have Canis, which is basically specific subfamily of dogs. And then within the subfamily of dogs, the organism is called Familiaris. So it's Canis familiaris. So can somebody tell me what is the common name for this dog by quickly Google searching?
Domestic dog.
It's a domestic dog, right? It's a house dog.
Yes,
All
sir.
right. So in GeneBank, you also have something called a feature table. So if you go back here, so I'm showing you the first part of the GeneBank record, and this is the second part of the GeneBank record. So you basically scroll down and you will see something like this. So you have something called feature source, right? Then you have something called CDS, which tells you from where to where you will have the protein in this sequence. So because it's an mRNA sequence, that means it's coding for a protein. So it says its N-terminal protein kinase domain starts from this location. It has a C-terminal myosin heavy chain head, and it's substrate for a pKa. So pKa is an enzyme. Then associated with this record, it also says, okay, this particular mRNA molecule makes this particular protein with its own accession, right? And then they can also give you the actual sequence of the amino acid that is found here below, right? So a lot of these gene records would also have this associated amino acid sequences or gene sequences that are there, and they will typically be shown in the form of a FASTA file or a FASTA file. It's a text-based format, and I've already told you there it can be the first line starting with the greater than sign tells you in some information about the identifier or an ID for the sequence, and then below that you have the sequence itself, which is typically stored in 80 characters per line. All right. So again, what's the role of public databases? Labs around the world send you data, they archive it, they classify it, they share data with others, they analyze it also, and they provide tools to help other researchers to access it and to analyze it as well. But in all this process, they are also knowing who is working with what kind of data. All right. So again, to come up with the primary and secondary database classification, primary databases, their main job is to archive, is to store. Secondary databases, their main job is to curate, is to become a knowledge base, is to have meta information about the original source. Then the source of the data is direct submissions from experimenters. Source of data for secondary databases is from results of analysis and basically interpretation of that data. Examples are given here for each one of them, and we're going to talk about them in detail. And the third kind of databases are called hybrid databases, or these are called families of databases. So they basically have features of both primary and secondary databases. That is, you can submit data, primary data on it, and also you can curate existing data on these platforms. And that's why it's more centric around one or few molecules that we'll be talking about. So for example, UniProt accepts primary sequences derived from peptide sequencing experiments, so basically protein sequencing experiments, but UniProt also infers peptide sequences from genomic information. So for example, if you were to give a gene's sequence, then automatically using algorithms, it's going to find the open reading frame in that gene sequence and corresponding peptide sequence. And then it's nowadays, because Google has released its protein sequence to protein structure software called DeepMind. What Google has done is they have collaborated with UniProt, and now from the sequence, you can also get a structure of the protein. So basically, you submitted your original peptide or DNA sequence, but this database, because of the tools which are available, it gives all this meta information as well. So it's almost like a hybrid version of the database. So
Sir, about Google's AI. So, sir, from one amino acid sequence, it gives just one tertiary structure of protein, or there are multiple. It gives multiple possible proteins structures. I just wanted to know.
it gives the most possible protein structure,
Okay, sir.
the
Okay, sir.
complete amino acid, given certain conditions. Okay,
Okay, sir.
so the primary databases that I have talked about, you can see, for example, our gene expression omnibus. This stores raw sequencing data from large projects generated using next generation sequencing platform. You have EBI's protein data bank, which stores 3D coordinates, or PDB. It stores 3D coordinates of protein structures, and it's associated with raw data from X-ray crystallography, NMR, and other techniques. You have secondary databases such as PFAM, or ensemble genomes, and they have hybrid databases such as UniProt or UCSC. So secondary databases can be feature centric. So PFAM essentially is a database for storing protein domains. So can somebody quickly tell me what a protein domain is?
It's basically a sequence of amino acids within the protein that performs a specific function. So a protein can
Performs a
have several
specific function,
domains,
and
like a binding.
yeah, and
Sorry, sir.
it's conserved across evolution, right?
Yes,
So
sir.
it's a specific sequence which performs a specific known function, and it's conserved across evolution. So I'm going to talk about each one of these domains in the remaining time, and each one of these database in remaining time. So for example, the first one, secondary database, is PFAM. So it's a protein family database. So proteins have one or more functional regions called domains, and the combination of different domains gives a protein the kind of function that it will eventually perform. So a PFAM essentially is a database that captures all this information. So for each PFAM entry, so if you will have a protein entered in PFAM, each PFAM entry is basically represented by a set of aligned sequences. So just like you had a motif matrix, you will have a set of protein sequences piled up one over the other in the form of a aligned sequence matrix. And this particular probabilistic, each one of these letters will then be converted into frequency space, and then from that frequency space it will be converted into what's called a hidden Markov model. If time permits, I'm going to talk about it as the last or the second last module of this course. But hidden Markov model essentially is another way of representing a greedy version of frequency matrix that we saw in the form of your motif searching. And given that you have a set of PFAM entries, like a protein family, which is represented like this, you can take this frequency matrix and you can search for proteins in a completely new organism based on this model. So related PFAM entries are grouped together as clans, and they are typically large and divergent superfamilies of multiple hidden Markov models or aligned sequences, where a single model cannot capture all the information, right? So it's pretty clear why do we need PFAM? Because we need to catalog different kinds of evolutionarily conserved parts of proteins which are performing certain functions. Now imagine in the future, assisted through all these deep learning technologies, if we are able to enumerate all the different kinds of domains which are present, or all the different kinds of small parts of a protein, you can imagine in a very near future, you can give or you can come up with an algorithm of making a specific design of a molecular machine. And just like engineers are designing machines using physical components, very soon in the future, even people are already doing that now, but in a much more larger automated fashion, given a specific molecular design, you can now, based on these protein families or based on these conserved domain information, you can actually put together domains and can achieve a function or you can achieve a structural version of a molecular machine that you would want to have. So PFAM families and clans. So there are basically 6 different categories, depending on the length and the nature of the sequence regions that are included in an entry. So the first entry is called the domain, smallest one, related sequences that form a distinct structural unit. You have a family. It's a collection of related sequence regions that may contain one or more domains. So typically you will see that 2 domains are seen quite frequently, so they form a family. Then you will have something called a repeat. So this is typically an unstable region which does not form a stable structure in 3 dimensions, but still could have some function. Then you have a motif. So it's a short unit that carries a distinct role. So for example, you have a metal binding motif. So if you have completed the case study that I asked you to complete in the lab yesterday, we were looking for a motif in the falciparum proteins, which are playing a role as a host factor. So they allow the malaria proteins to be exported inside the red blood cell and then cause the disease. So those specific motifs, for example, carry a very distinct role. Then there is something called coiled regions. So coiled regions of proteins which have alpha helices, and they align with each other to form these distinct structures. So they are different from domains and families because they are specifically a specific kind of, a structure, a secondary structure, which might have many distinct functions. One such functions is actually forming pores within the cell membrane to transport things in and outside the cell. And then there is, just like repeat units, you have something called disordered regions of the proteins. So these disordered regions of the proteins typically do not have any structure, but they are often enriched in lots of different kinds of motifs and these coiled regions sometimes that might but are sort of are conserved across evolution, because that's one of the major things that these this database particularly is categorizing. All right, so when you are specifically looking at PFAM, you can search for your protein or a DNA sequence in their models. If you write the name, let's say you write the name BRCA1, and they are going to tell you what all domains they could find in BRCA1, the domains that they have found, where all which all families and clans do these domains are a part of. And then specifically, you can go to each one of these entries, you can see alignments of these domains across evolution, across other different similar domains in other proteins, or same domain across different organisms. And you can know all this kind of information in this. Okay, so I'm going to stop here. In the class tomorrow, I'm going to talk about organism-centric databases. And here I'm going to talk to you about Ensemble. So a lot of the stuff that you have been doing, you in NCBI as a primary sort of database, you cannot just go to an organism and specifically look for things which are there happening in, let's say, human or mouse, right? And this is what Ensemble or organism-centric databases allow you to do. And we are going to discuss this in the next class. Are there any questions before I close?